Io adesso devo fare. C'è già. Dovrebbe essere a posto. Guarda un po'. Ok. Yeah, welcome everybody. Welcome everybody. We start my section now. I ask if you can please close the door. Okay, so I guess I need to sit because to change the slide, but maybe I also will walk a bit. So welcome again. I'm going to talk about population dynamics, or so-called population biology, specifically on corals. And specifically on the relation with climate change, but I will take in consideration for today the experiment that we did on ocean acidification. And we mainly work during a project, the Coral One project, where our colleague from Israel, also Jeb Kander, was involved from the 2010 to 2016. That was the official time of that project, but are still working on the output of coral worm right now today. And the main field work was the Panarea sites that you see these pictures. I will talk about this site a lot today. And there's a natural laboratory for studying the effect of acidification marine biodiversity, and we focus specifically on corals and on one of the aspects of corals biology that discuss calcification. And also we took in consideration mainly the reproduction. So that's my topic today. So I want to start for some basic information about corals ecology and biology. I ask if you can turn off the microphone, please. Thanks. I'm saying some basic aspect of coral ecology and biology. I know some of you actually are working on this organism, but others maybe are not so aware. So just few lines on this topic. Corals, of course, are organisms belonging to the phylum Cnidaria. The order is a Scleractinian, but that means Scleractina means a sea anemone with a sclero. Sclero is the skeleton. So basically, are sea anemones with that calcifying and exoskeletons. And they have two growth types, two growth form, solitary and colonial. Solitary species, solitary corals are living as autonomous individual. Each are autonomous from an energetic point of view. So you see here three modules, three organisms represented in this model here. Each of them represent a polyp. And you see how the polyp are actually not connected to each other. They are independent, autonomous from an energetic point of view. In this matter, in this sense, we are also solitary organisms, okay? Because we are not interconnected, we are independent from a trophic point of view. In contrast, we have the colonial growth form. Colonial organisms are interconnected. They are not only attached to each other, but they also share their gastrovascular cavity. So gastrovascular is the stomach, that in the cnidaria we call it coelenteron. So they basically are, the coelenteron are connected to each other. So this is a cooperative system. Means that, let's say that this polyp here on the top is eating something, is catching something through predation. When he's digesting stuff, he's also sharing his product, his energy to other member of the same colony. And another important point of the colonial aspect is that from a genetic point of view, they are, let's say, identical because according to the colonial model, the colony start from the founder, that is in this model is the polyp that is in the center of the colony. This polyp come from the fertilization of one egg. Okay, so the fertilized egg develop in the larvae that we call it planula. The planula at some point settle. The planula is the dispersive phase of the species. So it is a planktonic organism that is transported through current in the column water. And at some point, the planula settle, metamorphosize, and form the first polyp of the colony. That is in this model is this one in the center. And then from the founder, through asexual reproduction or budding, so there's no eggs or sperms in this case, but through asexual reproduction, through budding, the founder formed the second polyp. Then from 2 polyp, we have 4 polyp, 8 polyp, and so on, and we have the growth of the colony. So they are, in theory, genetically identical individuals that are interconnected and share their resources in a cooperative system. Then we have the energetic supply types. So we have basically, of course, these organisms are animals and are true predator. So they kill zooplankton. You know what is zooplankton? Are animals that are transported into the column water, and they prey zooplankton through their tentacles. And they kill the zooplankton animals through stinging cells that are the nematocytes. So in both type, you see left and right, we have the predation, but in some cases, actually, in the 50% of the species, not more than 50%, we also have established a symbiosis. So the coral polyp is not only a predator, a true animal, but is also active as a plant. I mean, it also get energy from the photosynthesis of the symbionts. The symbionts are named Zooxanthellae, that's the popular name, and they are dinoflagellate, unicellular organisms that are housed inside the cells of the animal, so in the cytoplasm. In the cytoplasm of the gastrodermal cells are the digestive cells of the coelenteron. Inside the animal cells, other than the ribosome, the mitochondria, the nucleus of all eukaryotic cells, we also have the presence of many dozens of ZOCs, of these microalgae, unicellular microalgae that are hosted inside the cytoplasm of the animal cells. And they, of course, are photosynthesizing organisms, and they share a great part of the energy that come from the sun, from the photosynthesis. They translocate this these sugars that come from the photosynthesis. They translocate this organic matter into the metabolism of the animal cells. So they are of a great support of the metabolism of the polyp. There are a few studies that try to estimate the amount of energy that from the photosynthesis go into the animal metabolism, and in some oligotrophic areas, like in the warm tropical sea of the Pacific Ocean, up to 90% of the energy that the animal cells need for its own metabolism come from the photosynthesis. So the photosynthesis is very important in the zooxanthellate species. So we call zooxanthellate species or zooxanthellate corals the one are hosting the zoox, and non-zooxanthellate the corals that are not hosting this symbiosing. So we are talking about corals now, and this is a peculiar place, as I said. This is the Panarea crater. We started working on this place in 2010 after we got a grant together with Zvi Dubinsky, a professor of Faculty of Life Science of Bar-Ilan University. I get a bit moved now because he passed away last year. Unfortunately, his name is Zvi Dubinsky, you see there. And then there was a second group from Bologna that was coordinated by Giuseppe Falini of the Department of Chemistry and of myself. So there were two groups basically, one in Bar-Ilan and one in Alma Mater Studiorum, our university. And the grant was from the ERC, the European Research Council. I suggest you, I know maybe some of you already know this funding agency. I suggest you to look at the ERC when you have time, because it's the most important agency supporting basic research in Europe. And they have specific program for the postdoc. So after you graduate as PhD student, then right after, you may have the chance to compete with all the other postdocs in Europe for getting this prestigious grant. I'm sorry, I have this cold that is killing me since December. And in this case, the grant was for advanced, old people, okay, but they have a special grant for the young people like you. So please visit the ERC website. Yes, I said this is a special place. I will describe you later the details. Basically, why it's a crater. It's an underwater crater. It's in Sicily, in the North Sicily. Actually, there is in this small archipelago, the Aeolian Island, that is characterized by a volcanic activity. And in this specific site in Panarea, the gas emission, in contrary to the other place around the Aeolian and around the world where the volcanoes emit any kinds of toxic compound, acidic, like a dangerous compound for the biology. In this case, the emission are only, there's 99.9% of carbon dioxide. Anidride carbonica, CO2, okay? So basically, the water here are acidified to the level expected by the end of the century with the same mechanism of climate change, that is carbon dioxide. So in this place, you can try to infer what are the potential acclimatization process of the organism in the future climate change conditions, specifically for the ocean acidification. So we are now. So the topic, the denominator of my presentation today is the ocean acidification. You are all aware. This is stuff that you saw many times. So I don't want to stay here very long. You see since 10,000 years ago, but we can go back in time with the geology and the geochemistry data much more back in time. You see the carbon dioxide concentration in the atmosphere that is slightly homogeneous. I mean, it's slightly changing, but it's basically homogeneous over 10,000 years, the last 10,000 years. Then at time zero, which is today, you see this vertical increase in carbon dioxide. And this vertical increasing is basically starting from the second half of the 19th century, the second industrial revolution, where we start using fossil fuel as a main source of energy in our society. So because of this carbon dioxide emission, we have an increasing in temperature, the global scale, global warming, and then because of this, we have a global sea level rise, decrease in snow cover, and so on. When we wrote the coral worm, the proposal, it took us more than 2 years. So we start in 2007 2006, 2007 in drafting this proposal. I can tell you that at that time, the concept of ocean acidification was not so diffused in the scientific community either. We were well aware of global warming, but I will show you one of the first scientific paper in Science was published by Hove Goldberg from Australia during this period, 2009, 2010. So in coral worm, we did not focus. In fact, the acronym is coral warming, and not coral acid, because the scientific community comprised ourselves were not so well aware of the acidification process. Then the ocean acidification. The carbon dioxide does not remain in the atmosphere, but for a simple physical law, the Henry law. The higher is the partial pressure of the carbon dioxide in the atmosphere, the higher is the dissolution of this gas in the ocean. So, and you see here one of the many databases that you can find online or published in many papers about the relationship between carbon dioxide in the atmosphere on the top and the pH trend at the bottom. This is an oceanic buoy from the University of Hawaii. This is in the middle of the Pacific Ocean, and show you along a number of decades the increase in partial pressure concentration of carbon dioxide in the atmosphere, and the correlated decrease in oceanic pH. So why this is happening? Because the CO2, when it's entering into the water system, it immediately reacts with water, producing carbonic acid. The carbonic acid is a weak acid that immediately releases one proton, these hydrogen ions. So the higher is the carbon dioxide in the atmosphere, the higher is becoming the concentration of these hydrogen ions in the ocean, and the lower is becoming the pH. So here there are some data around that years. You see, this was 2005, more or less. They start reducing this prediction about the ocean acidification. This is a paper in Nature, and show us how the pH will change in the next future during this century. From pre-industrial means before the second industrial revolution, that was around between 8.1 8.2. Today we are here around 8.1 is the average oceanic pH, and then at the end of the century the pH will drop down to a few more decimal, down to 7.9, 7.7 in the worst case scenario. So this may look a little change, especially for people working in molecular biology, or if you did some experiment during your period at the high school in the lab, you were for sure working with a solution in the labs that go from pH 9 to pH 2. So it looks like a very small change, a few decimal. But if you need to remember that the pH is a logarithm transformation of the actual concentration of hydrogens. So if you transform the logarithm into real concentration, and then you calculate the percentage of change between 8.1 to 7.8, it means that the concentration of hydrogen ions in this very short time will increase of around 100%. So the sea will become double, the 100% more acidic that was during the last many thousands of years. So many organisms are calcified organisms. Calcium carbonate, I can say, we have here expert in biomineralization, Frederick Morin. You just had the lesson yesterday. I couldn't attend, unfortunately, because I was busy with some institutional appointment. But of course, you understood that the calcium carbonate is the mineral that the natural selection selected for most of the organisms, from the plants to the animals, from the metazoa to the unicellular organism. When the organism needs to produce the skeleton, okay, the mineral that was selected by the natural selection was the calcium carbonate. We have few examples of skeletons made of other kind of minerals, but there are few cases compared to the calcium carbonate. So we have the organisms that are calcifying, are producing skeletons or structures made of calcium carbonate. And we have some example. This is from Caldera Oceanography. There's a very important paper that show the different calcifier. So we have a calcifier in the marine environment that are pelagic. Some, for example, foraminifera here you see, and these are pteropods that are mollusks. And then coccolithophore, these are microalgae. And these are pelagic organisms. And then, of course, we have a calcified organism at the benthos domain. And of course, we have corals, that is the subject of this lesson, sea urchins, and many, many other organisms. So calcium carbonate, the classification is very important for the biology in general. There is another classic paper. This is from people from Monaco. This is an estimation of the calcium carbonate global production. You see here by the major marine calcifier. So you see this graph here. So we have the forams as a major calcifier at the global scale. In the first place, and then we have a coccolithophore, this microalgae, and then we have corals that have more or less the same importance as calcifier organism as echinoderms here, and then you have the mollusks and so on. Basically, they estimate that in total, the amount of calcium carbonate produced per year by the calcified organism, it's around 5 milliards of tons per year of material. 5 million of tons of material looks like a big number, but maybe we need to understand what does it mean, because maybe we are not really very well aware of the importance of calcification. I was last October in Poland to a geological congress organized by our colleague Jaroslav Stolarski. And I had some field work with the geologist. I was still really impressed when I think what does it mean, the calcification in terms of amount of material, substrates that they are able to produce. So let's try to see this a bit more with some example. So these are structures that are the results, the output of calcification, or we also like to talk about biomineralization, because we want to focus on the fact that this calcification process is biologically controlled. There's a lot of geochemistry, chemistry, but there's the importance of biology. It's irrelevant. So this is the importance of the biomineralization in terms of structures. This is the GBR, the Great Barrier Reef, is in the east coast of Australia, is thousands of kilometers long, and is providing a very important ecosystem services. If you want to put this idea on the practical sense. They protect the coastline from tsunamis, they sustain fisheries, they sustain important economic sectors, not only fishery, tourism, and this is the and many other ecosystem services. The same if you look at the atolls in the Maldives, in the middle of the Indian Oceans. But still, this stuff are things that are very far away from this room. Okay, so looks okay that yeah, it's important, but it's there. It's not here. It's there. Because let's go closer to this room. This is exactly from the Adriatic. This is the coral reefs in Croatia, in the Mileto Island. It's just in front. We are in Fano. If we take a boat, a few hours across the Adriatic, we arrive in this island where there is these coral reefs that is made is built by Cladocora cespitosa, that is an endemic species of our Mediterranean Sea. And this is a reef, an entire reef that it's, of course, as ecosystem services, as a hot spot of biodiversity, and so on. This is Lophelia pertusa. Now they changed the name, the taxonomy is in Desmophilium pertusum. Is a cold water coral living down thousands of meters below the surface in the dark, in the abyss, and it's forming this huge reef, again, hotspot of biodiversity and sustaining important ecosystem services. And this is these are the Alps in the North Italy. These are biogenic structures. They are made of shells, echinoderms, corals, and these are an example of the importance of this biomineralization process. So the biomineralization is actually producing the substratum where we are actually living, walking, building our city, our infrastructure, and maybe also vacation. So these pictures give you an idea of what does it mean 5 million tons of calcium carbonate per year. Okay. So what is the relation between this biomineralization and ocean acidification? Is that the acidification, in theory, could affect negatively the potential calcification process of this organism. We know a bit more than 15 years ago about the ocean acidification. There's a bunch of people working during this year on this problem to try to understand what is actually happening and what is the acclimatization potential of this organism in a more acidified environment. So why the acidification is as a problem for the production of calcium carbonate? This is one of the first paper, as I said, that was published, you see, in 2007. And basically is the most important, one of the most important paper explaining on giving a general view of the problem of ocean acidification for calcifying organisms. This is the paper that I mentioned you at the beginning of my speech today. It's about Hof Goldberg, a scientist from Australia. He's also a member of the IPCC, the International Panel of Climate Change. Is very well known as an eminence as an authority in this field. But this was just 2007 and was published in Science. You know this journal, I don't need to present. And through this cartoon here, is explaining the problem of ocean acidification. From this paper and a few other papers, but I want to say from this paper, people start looking more closely to this potential problem. So what's happening? We are producing carbon dioxide, of course, and the carbon dioxide reach the atmosphere. This is increasing concentration carbon dioxide, is increasing the temperature, the global warming. But then there is Henry, this guy that studied the diffusion of gas into the water, for example, into the fluid, that explained that the higher the partial pressure in the atmosphere, the higher is the diffusion of carbon dioxide into the water. So the more CO2 we produce, the more CO2 enter into the ocean. As I said before, the carbon dioxide immediately react with water, producing the carbonic acid that is releasing one proton. Okay, so this increasing in hydrogen ions concentration is decreasing the pH. So the oceanic pH is decreasing. That's what you are measuring. Then the hydrogen ions is a positive ions that is looking for a negative ions. Cannot stay alone like a positive. Okay, it's not in equilibrium. So in the oceanic environment, the water environment, he immediately react with the carbonate ions that has two negative charge here, and the reaction is producing bicarbonate. So this one is the bicarbonate ions, and this is the carbonate ions. So the acidification basically has the results of removing the carbonate ions. The carbonate ions is the one is reactive with the calcium in order to produce calcium carbonate. So for the gasification, you need the calcium, you need the carbonate. The carbonate is removed by the environment because of acidification. So this means that it's becoming more difficult to produce, in theory, more difficult to produce calcium carbonate in an acidified environment. Okay, not only, but when you see here, there are all reaction at the equilibrium in theory. So if the concentration of carbonate ions is decreasing, okay, this is potentially leading to dissolution. This is calcium carbonate. You see here coral, calcium carbonate. This is calcium carbonate that is already mineralized, okay? And this calcium carbonate can dissolve into the water, okay, in order to restore the oceanic equilibrium. So with acidification, we have two problems. First one, the decreasing in carbonate ions that make more difficult the gasification process. Second, the dissolution, the potential dissolution of calcium carbonate, means skeletons that already are formed, in order to restore the oceanic equilibrium. So these things was very well explained in this paper in 2007. So this is another way to explain the problem of acidification from a poor chemistry point of view. Here there is no biology at all. So this is the omega aragonite that basically is explaining the saturation state of calcium and carbonate into the water environment. The higher is omega, the more saturated is the environment, the higher is the probability of producing calcium carbonate. The higher is the precipitation. That's from a thermodynamic point of view. The higher is omega, the higher are the concentration of calcium and carbonate, the higher is the probability of precipitation. The easier is for the precipitation of new calcium carbonate. The lower is omega. When omega becomes lower than 1, I mean that the environment is undersaturated. So it's difficult, the production of new calcium carbonate, because the ions cannot interact with each other, okay? And in the undersaturated condition is favored the dissolution. Dissolution means the one I explained you before, that the calcium carbonate that is already mineralized tend to dissolve into the environment. So for calcification, we need a higher omega value. The higher is the omega, the easier is the precipitation of calcium carbonate. This is from a chemistry point of view. There's no biology here. And then we have this product of solubility K that is depending on pressure, temperature, and salinity, but I'm not going to go in this detail today. But this K is not a constant, but it is a product. It's the product of solubility that depends on temperature, salinity, and pressure. Okay, so in theory, in the acidic environment, in theory, the organism should have problem for the classification process. That's the problem. But the classification process is happening in this little space here that is at the bottom of the polyp. Here you see the scheme, classic zoological scheme of anthozoan polyp that show you how the polyp is organized. So basically, we have this organism that is as an empty sac in the center of the body. This cavity here is the gastrovascular cavity that we call coelenteron. Okay. Then we have the tentacles, of course. The tentacles, they use the tentacles for preying, to prey the zooplankton. They are real predators. Zocs, non-zocs, they are predator. And then we have in terms of cellular layer, they are diblastic organisms. It means that they only have two tissue, real tissue, that is the endoderm and the ectoderm. The endoderm is, or it's better indicated as gastroderm in the full differentiated polyp. The endoderm or gastroderm is this line here, is the one, is delimiting the gastrovascular cavity. So we have the endoderm. And then we have in this layer here that is an acellular layer that we call it mesoglea, is rich in proteins, but is not cellular, is not a real tissue, is a kind of gel, is a gel that is separating the endoderm or gastroderm to the ectoderm. This continuous line here is the ectoderm, okay, that in the adult, in the developed organism is accurate to indicate as epidermis. So they have only two layers, very, very easy the organization. So again, the problem of acidification is happening here in the ocean, okay? But the calcification process, as I will show you in a few slides, is happening here, is occurring here, in this space that is in the aboral side, of the polyp. We call it this oral pole, oral, because this is the mouth, oral pole, aboral pole. At the aboral pole is the place where the calcification is occurring. This is another famous diagram from Veron that we use in our book to explain some concept. Again, the ocean acidification is happening here at this scale, at the oceanic scale, up here. But here you see, again, the diagram showing you the epidermis or ectoderma, the green one, and then the mesoglea. This is not a tissue, okay? It's a gel, it's a not cellular layer. And then we have the gastrodermis. You see the red one, that is the tissue that is delimiting the gastrovascular cavity. And then at the aboral pole, okay, there are the same tissue. We have the gastrodermis in red, the mesoglea, then you cannot see here a very thin layer of epidermis at the aboral pole. This very thin layer of cell, of epidermal cell at the aboral pole are the ones that are in charge of calcification. But I want to stress again the spatial scale of the problem. We are talking about ocean acidification here, okay, and the calcification process in this micron space here at the aboral pole. This is another diagram from, again, people from Monaco, Tambouté et al., that show us how the polyp is organized. Okay, again, the seawater, the ocean up here. And then we have at the oral pole, we have ectoderma, the ectoderm here, or epidermis, where there are the nematocytes, the cells that are used for depredation, the stinging cells. The layer of mesoglea, this is acellular layer, okay, rich in proteins and other molecules, but it's not cellular. And then we have the gastrodermis or endoderma of the oral pole. And in the endoderm or gastrodermis of the oral pole are hosted, you see here these big cells, the zocs. Okay, the zocs are mainly distributed in the gastrodermis of the oral pole because they need to catch light for the photosynthesis. Then we have the coelenterol, the gastrovascular cavity, and then at the aboral pole, at the base of the polyp, again we have the same three layer, the cellular layer that is the gastrodermis here, the mesoglea, this is pink here, and then you see this thin layer of cells that are the calicoblastic cells that are in charge of the calcification. And then the ECM, the extracellular medium, in orange here, you see this is a fluid. There's a fluid that is in between the ectoderms, okay, and the skeleton. So these calicoblastic cells, these ectodermal cells that we call it calicoblastic, calico because means they produce the calcium carbonate, they calcify. These calicoblastic cells, these epidermal cells, are the one that produce the skeleton, and you see the skeleton is an exoskeleton. The polyp, as you see here, is actually sitting, literally sitting on its own skeleton. It's not inside the body. It's an exoskeleton produced outside the body of the polyp. So the polyp is actually sitting on his skeleton, and you see here this magnification, how the calicoblastic cell are producing the skeleton that is at the base of the polyp. And this extracellular medium in orange is the medium where the calcification process is actually taking place. And there is a lot of discussion in biologists now about this, the characteristic of this extracellular medium. There are some colleagues that basically believe that is fundamentally seawater that is getting to this space. Other that think that this medium is very modified by the metabolism of these cells. But anyway, this extracellular medium is the place where the calcification process is taking place. So this is another model. This is a model from Kuif and Dauphin, 2005, Biogeoscience. It's a classical model of biomineralization that is explaining how the coral skeleton is produced. And is produced by the formation of layers. There's formation of one layers, and then another layers, and then another layers, and so on. We need to read this scheme from the bottom to the top. So we start from, you see, A, B, C, D. Okay, so let's start from the bottom to read it. So we have, you see here, we have already 3 layers of skeleton that already formed. And the calicoblastic layers, the ectodermal cells here. So according to Kuif and Dauphin model of biomineralization, we need to produce another layer, okay? And so layer by layer, the skeleton is produced. So the first things that the ectodermal cells do for the formation of the skeleton is the production of the organic matrix. Probably Frederick already explained you on Monday. The organic matrix secretion. So externally, okay, they produce this framework of macromolecules. This organic matrix is made by the main kind of biological molecules, proteins, lipids, then carbohydrates. So because these molecules are amphipathic, that means that they have some part that are more hydrophobic and other that are hydrophilic. It means some part of the molecules, they like water, other part of the same molecule doesn't like water. They have a specific oriental disposition in the space, like all the proteins and all the macromolecules. Within this framework, it's occurring the crystallization, the reaction between calcium and carbonate. And this is the next phase. The calcium carbonate crystallization is occurring inside this organic matrix. That we believe the organic matrix has also a role in the morphology of the skeletons. The fact that shells of gastropod as a shell that is completely different from a bivalve, or one species of corals has a specific morphology different from another species in their skeletons, also depends on the role of this organic matrix at this level. So then you have here the role of magnesium. I need to mention it very briefly, without going into the detail. We know that the magnesium has a role in the type or polymorph of calcium carbonate that is produced. The skeletons are calcium carbonate, but you know probably that there are different polymorphs. Two main polymorphs are aragonite and calcite. It's always calcium carbonate, but these two kind of calcium carbonate have a different density, different orientation in the crystals, so they have some different of the crystal scale. And the magnesium has a role in favoring the precipitation of aragonite. The modern coral have the skeletons that is made. The bulk of the skeleton is made of aragonite. Okay, it's basically aragonite, calcium carbonate in the form of aragonite. That's the skeleton of modern corals. So there is the role of magnesium in this model. And from here, we go to the last step, the formation of the next layer. So you see, we have 3 layers, and then we arrive to 4 layers. And the last things that I need to say before going into the experiment that we perform is this is from Cohen. It's a US colleague, is explaining the physiology. There are many models on this physiology, but more or less with some difference, the basic message that they give us is that to produce calcium carbonate, the reaction between calcium and carbonate, we need both ions, okay, for sure. And the higher is the pH, okay, the higher is the concentration of carbonate, and the easier is the precipitation of the formation of new calcium carbonate. So we need high pH in order to have the calcification. In this specific model, Cohen and colleagues, they show us the calcium ATPase, that is an enzyme pump that has the role. There are other pumps for sure. They put their attention on calcium ATPase, but anyway, the meaning is that this pump has the role of removing hydrogens from where? From the calcified fluid. Okay, this is the calcified fluid is here in this little space where there is the production of the organic matrix and where there are the physiological reaction that lead to the production of calcium carbonate. We are talking about this space now. So that enzyme that pump, that protein, is increasing the pH here in this fluid here. So we are here. So the meaning is that they need to remove hydrogen in order to raise the pH, to increase the pH. For each 2 hydrogen ions that they remove, this calcium ATPase pump is releasing inside the fluid one calcium ions. But as I said, the meaning of this pump is not to increase the amount of calcium in the fluid, because in the ocean and also in this fluid, the calcium is not a limiting factor. The limiting factor in the calcification is always the carbonate. So you see here, if you remove the hydrogen, as I explained you before in this equation here, okay, if you remove the hydrogens, you increase the concentration of carbonate. So the meaning of that pump is removing hydrogens in order to increase the concentration of carbonate. And to do this job, that's another important point, is that there is a TPA, so it needs energy. So in order to remove hydrogens, okay, to do this work, it needs a lot of energy, a lot of ATP. So this pump is raising the pH, the carbonate ions becoming concentrate, react with the calcium, and then we have the production of calcium carbonate. So the problem was, and he is today still, even if we understood something, but not really complete pictures, as you will see, is the acidification and how this is affecting the gasification potential. So we, of course, we did some work in the lab, in the control environment, but most of our work during this 15 years, 16 years, is coming from the field, and a great part from this pan area. So what we did there, we did many things. I'm not going in all the experiment, of course, and studies. I focus on some aspect. One of the approach that we try to realize there is a transplant experiment. We transplanted 3 species of corals along a pH gradient that I will show you in a few seconds. The 3 species were this one, Balanophilia europea, that some of you know very well because it's working on that. Balanophilia europea is a ZOX solitary coral, so it's a solitary polyp. You see here's an individual, solitary, not colonial means, and is a ZOX. Is a zooxanthellate, means that a part of the energy for its metabolism is coming from the photosynthesis of the symbionts. And is the only one ZOX species that we use in this experiment, in this transplant experiment. Then we used as a model species Leptosamia pruvoti. Years ago, Leptosamia pruvoti was the name was Balanophilia pruvoti. This means that a very close species to Balanophilia. The main difference from Balanophilia europea is that Leptosamia pruvoti is a non-ZOX. So it's a non-ZOX corals. And then we use as a third species Astroides calicularis. Astroides calicularis is a colonial coral, as you see here, non-ZOX. So we have 1 ZOX and 2 non-ZOX species. And they all belonging to the same family, the Dendrophilidae, so it means that they are very close phylogenetically. So there is not a phylogenetic bias in the response that we have, because our goal was to compare the acclimatization potential of non-ZOX against ZOX. Okay, and this is the comparison between Leptosamia and Balanophilia, because they are both solitary, but one is non-ZOX and the other is ZOX. So the main difference is the presence or absence of photosynthesis. And between colonial and solitary form, that is the comparison between Leptosamia pruvoti and Astroides calicularis. They are both non-dogs, but one is colonial and the other is solitary. So what we do in what we did in Panarea. So Panarea, it's this island here. So we are in the South Italy, in the north coast of Sicily, out of Sicily, in the north, out in the north here of Sicily. There is this archipelago, the Aeolian archipelago. The Aeolian archipelago is basically this island here are the remaining of a volcanic crater. Okay, and all the area is still very important from the volcanic activity is still very important. In fact, we have Stromboli here, the northern island is Stromboli, that volcanologists consider the most active or one of the most active volcanoes on Earth. And then a few kilometers south of Stromboli, we have Panarea. So all this area is characterized by the vents of gas from the volcanic activity. When volcanoes produce its own gas, the gas has a reach of any possible kind of toxic compound, very acidic in some many of them. But in a few cases, Panarea is one of these. The volcanoes, for a mechanism that the geologists know to explain very well. In this point, the emissions are basically carbon dioxide. In 99.9% of carbon dioxide, of CO2. We are here, so Panarea, in front of Panarea, this little island, with now in this period, we are in February, and we are close to Carnival, and we were there many years ago sampling, and in this period in Panarea, there are 25 people living on the island. In summer, instead, is one of the main attractive area, very, very important from the touristic point of view. But in the winter, there are fewer inhabitants there. So here is the jetty, the little village. In front of the little village, there are a small a group of small rocks that the island people call islands, but are very little piece of rock in the middle of the Med. And you see here, and one of these rock is Bottaro, and west of Bottaro, 50 meters from Bottaro, there is the crater, the CO2, the gas in place. So that was the place that we discovered through a geologist that is also not here anymore, had an accident some years ago, Bruno Capaccioni, and he was the guy that discovered this place and was following the evolution of this place many years. And we were introduced by Bruno in this crater, Bottaro crater. Nobody was working in Panarea. We started working there, doing the experiment in the ecology or biology matter. It was only followed by this small group of geologists. So now I want to show you the experiment setup, saying that this setup that you see in this video is not the one that produced the data, because this setup was completely destroyed. We took 1 year of we took 1 year and different experiment just to understand how to set the experiment underwater, because it was destroyed the first 3 times, the first 2 times completely, the third one almost by the waves, because the ocean during the winter here is very strong. So you need to set the tables and everything, not in this way, but it still give the meaning of the things that we did. So we are here basically 30 meters away from the center of the volcanic activity. You see here the setup. You see here the Astroides calicularis. Astroides calicularis is the colonial non-zox corals. These are piece of colonies, nubbians, piece of colonies. The colony is able to colonize caves, of course, inside caves, but also in Panarea is very abundant also outside the caves in the sunny habitat. So we collect the corals from a sunny habitat in case of Astroides. So that's the reason you find these non-zox corals in this position towards the sun, because we are mimicking the same position from where the corals were collected. And then close to Astroides, there is Balanophilia. These are Balanophilia polyps. They are, of course, toward the sun, because this is a zox coral. It must stay under the sun. So we are mimicking the position, okay, the natural position. And then in the shadow habitat, in the shadow part of the experiment, you see Leptosamia pruvoti. Leptosamia pruvoti is a solitary non-zox, is only living inside shadow habitat in caves, cavern. So we collect the corals from caves, and we attach the corals in a shaded habitat to mimicking its natural position. Here we are, more or less, I said, 35 meters away from the center of the crater. The crater is there, is down there, and now we are swimming toward the crater. You can see here, 35 meters away from the center of the crater, you see Posidonia oceanica, that is this plant, that is one of main ecosystem in the Mediterranean. So it's a normal Mediterranean ecosystem here. You can see some fishes, for example, here swimming. So 35 meters away from the center, the pH is normal, is 8.1. And we have a normal ecosystem, typical Mediterranean ecosystem. So now we are swimming toward the center of the crater. The pH is decreasing. And you see how suddenly the Posidonia disappear, and the community become dominated by this macroalgae, different species, but there's no Posidonia anymore. And this is the site 2. Okay, the first one was site 1 in the Posidonia in the normal condition. Then this is site 2 in acidified water. This is the crater here. Look at here. This is the crater. This is site 3. Site 3, and this is site 4. Site 4 was very close to the center of the crater, where there is the main carbon dioxide emission. Here, you see site 4, and this is the carbon dioxide emission. And this is carbon dioxide, as I said, 99.9%. The presence of other toxic compound are not detectable by the instrument. So there may be some sulfur around, but it's very, very low, the concentration. So this means that the environment here is acidified through carbon dioxide with the same mechanism of climate change as I explained you before. And this is comparing the pH of the experiment with the pH predicted by the end of the century. So this is site 1. In site 1, you see the distance. Here you can read the distance from the center of the crater, and in the y-axis you have the pH. So 35 meters away from the center is a site 1. The site 1 has a pH of 8.1. That is the expected pH for the ocean today. So it's a normal pH. In site 2, we have a pH of 7.9, between 7.8 and 7.19. And if you look at the scenarios predicted by the end of the century here, this is the IPCC scenarios. You know, the IPCC scenarios, they always produce different scenarios according to the possible policy of on mitigation of climate change. Okay, from the more optimistic, that is this one, that unfortunately is not realistic because we are not implementing nothing for mitigate climate change. So this is the most optimistic, and then we have intermediate scenario, and then we have the pessimistic, the most pessimistic scenario that is defined as business as usual. That means that we are producing carbon dioxide without any mitigation. So we can compare the pH of the experiment in the field, thanks to the Panarea crater, with the pH predicted by the end of the century. If you look at site 2, for example, we have a pH between 7.9 and 7.8. That is exactly the pH predicted by the IPCC for the end of the century. So today in Panarea, that side, 50 meters away from the crater, we have the ocean that, according to the IPCC scenarios, will be the ocean at the global scale at the end of the century. For the intermediate scenario, in site 3, we have a pH of 7.7 that is corresponding exactly to the pH predicted by the IPCC for the end of the century, this century, for the most pessimistic scenario. So site 3 has today the condition that the global ocean will have, according to IPCC, for the most pessimistic, according to the most pessimistic scenario. In site 4, we have a pH of 7.4. Look, 7.4 that is not predicted by the IPCC scenario. So the pH is lower than the most pessimistic prediction for the end of the century. So pH 7.4 we consider a pH that an extreme environment. Okay, because the water is more acidic than most pessimistic scenario according to the IPCC. So now what we did, we transplanted the corals along the gradient. We studied the regeneration rate. There's a paper of students, your colleague here, where is? La. And we studied the regeneration, Teresa, the regeneration, the calcification, the mortality, many. Today I'm gonna explain just the calcification data. These are the raw data. Then, of course, we produce this nice 3D model in the paper that was published. But I prefer to show the raw data that I think are more useful to explain the results. So this looks like a terrible table, but it's not so difficult to read. We have 3 lines. In each line, we have one species. The first line is relating to the ZOX coral, Balanophilia europea. The second and the third line are the non-ZOX corals. This one is Leptosamia pruvoti, that is a solitary, and then we have Astroides calicularis, that is the colonial one. And then we have 3 columns, one for each season. We repeat after we fail in 1 year, okay, because of the waves and the destruction of the experiment, we repeat the experiment many times in different seasons. Each experiment, as you see here, was lasting about 3-4 months. So we have column here. Column, this was the experiment in cold water between November and March 2010-2011. You see here the temperature. So this is a cold water experiment, an intermediate temperature, and the warm water experiment. And then we have in each graph the calcification of the transplant corals in the y-axis and the pH where the calcification was measured. So we have the corals are site 1, the corals are site 2, the corals are site 3, 4, and how they calcified during that experiment. So if you look at the first line, Balanophilia europea, the Zox coral, the only Zox coral that we have in this experiment, you see that there is, for example, in the cold season, we have a lot of variability, okay, in the calcification, up and down here, but there is no difference among sites. Okay, there is no difference among pH condition. Okay, the same things we have in the intermediate temperature. The variability decreases a lot. You see here there is less variability, but still there's no variation among the pH sites. And then in summer, there's a variation, but it's not related to the pH. So in Balanophilia europea, basically, we did not find in these 3 experiments and in all the other experiments that they were then modeled in the nice 3D modeling, okay, we never found any relationship between pH and calcification in all the temperature that we tested. If we go in Leptosamia pruvoti, the second line, non-Zox, again, we have in the cold season bunch of variation, no difference among sites, a decreasing in variation in the intermediate temperature, but still there's no relationship with the pH. In the summer, in the warm water, okay, in warm water, there is we found a significant difference in the calcification rate among sites that was related to pH. So in summer, there is a calcification that is negatively related to pH. Okay, the lower is the pH, means the higher is the acidified environment, the higher is the acidification, the lower is the calcification rate. But we found this only in the warm water, not in the intermediate and the cold water. So there is a kind of synergistic, maybe we can use this adjective, effect between temperature and acidification. In Astroides calicularis, the last line, that is the non-Zox and colonial, in all the season tested, we always found a relationship between calcification and pH, where the calcification was lower was lower in the more acidified site. So the higher was the acidification, the lower was the calcification in all the season. In the last line, we can put our attention in this raw data, very easy elaboration, in the intercept point of the regression line. The intercept point is the point that is separating the positive calcification from the negative calcification. Negative calcification means that the weight of the skeleton is decreasing. So it means that there is dissolution. Okay. So this regression line is actually dividing the sector in 2 sectors. Positive calcification, that means that the skeleton is actually accumulating calcium carbonate from dissolution. And if you look at the intercept point, actually I can ask you, according to your eyes, the intercept point is always in the same position or is moving with the season? It's moving toward higher pH. You see? It's here in winter, and then goes here in the intermediate water, and then more on the right in the warmer water. So it means that the higher is the temperature, the higher is the temperature, the higher is the intercept point. Means that the higher is the temperature, the higher is the dissolution. Okay? In cold water, in order to dissolve the skeleton, the pH must go down to 7.9. so needs acidified water. In summer, it's just below 8.1, you already start having a dissolution of skeleton. So again, in the non-ZOX corals, also in the last case, we see an interaction, a synergistic effect between temperature and acidification, where acidification is where the negative effect of acidification is amplified by the temperature. The ZOX coral does not react. I mean, you see here, no matter what is the temperature, always the calcification looks like not affected by acidification. So the ZOX coral look more resistant than the other one, okay, in this experiment. And we try to explain this with different hypotheses, mainly three. One is the role of photosynthesis. The second one is the role of the organic matrix. And the third one is the homeostatic capacity of the corals. Now, start from the photosynthesis. I'm going to explain you these three hypotheses that we use to explain the results, and then we have a brief stop. So first, the role of photosynthesis. The ZOX corals is more resistant in this experiment. That's for sure. In this experiment that was lasting 3-4 months, the ZOX coral was more resistant than the non-ZOX. The first is the role of photosynthesis. Why? Because if increase the carbon dioxide, which also with acidification, we saw that the calcification, if increase the carbon dioxide, and increase the acidification of the environment, the calcification has a potential problem. We explain here at the beginning of the speech. So acidification is making problem to calcification. But if the coral is a photosynthetic corals, you know, carbon dioxide is the substrate of photosynthesis. So more carbon dioxide may have a positive effect on photosynthesis, maybe increasing the photosynthetic production or increasing, as we will see later, the number of ZOX. Anyway, the carbon dioxide as a substrate of photosynthesis may have a positive effect on photosynthesis. That our hypothesis that was tested in another experiment that I show you later. More photosynthesis means more energy, because more photosynthesis is more organic carbon that can be used substrate for respiration, the production of ATP. ATP is essential for the calcification, as I mentioned you before. So in this ZOX coral, the negative effect of carbon dioxide on calcification may be balanced by the positive effect of photosynthesis, and this is maybe giving an explanation of why the calcification is not affected in this enriched carbon dioxide environment, because the coral is suffering in one side, but may have a positive effect of the other side, and there may be a balance between the positive and negative, keeping the calcification process homogeneous. The second one is the organic matrix story. This is a paper that we published already, was 14 years ago. You see here the reference. And the expert in this organic matrix in Bologna, Frederick is a super expert, as you know. In Bologna, there's Giuseppe Falini. We did this together, Giuseppe. And what we did is that we extracted the organic matrix from different species. The tree of the experiment comprised Balanophylla europea, of course, and other species from the tropic. And we did some in vitro experiment. We were manipulating the concentration of calcium, of magnesium. So basically what we did, we took the skeletons we extract from the skeleton the organic matrix. We put the organic matrix in vitro environment, artificial seawater, and then we manipulated the amount of magnesium in the environment. And we did this for a number of species. I mentioned you before that the magnesium is the element that is boosting the production of aragonite. So this means that with high magnesium, the calcium carbonate is in form of aragonite. In low magnesium, the calcium carbonate is precipitated in form of calcite. So where the magnesium is high, is favored aragonite. Where the magnesium is low, is favored the calcite. So we manipulated the amount of magnesium in the artificial environment, bringing the concentration of magnesium in artificial seawater down to zero, no magnesium. So from a thermodynamic point of view, the expectation was that all the organic matrix would produce calcium carbonate in the form of calcite, because no magnesium. And in fact, they didn't. All the species produce calcium carbonate in the form of calcite, except one, Balanophilia europea. Balanophilia europea, even in the absence, complete absence of magnesium, was able to precipitate calcium carbonate with a significant amount of aragonite. All the other corals were not able. So this means maybe that the organic matrix of Balanophilia europea may have some peculiar characteristic leading to a more controlled environment at the calcification site, as a stronger biological control of the calcification process compared to the other species. And this maybe could happen also in the acidified environment. So one second hypothesis is the role of this special organic matrix of Balanophilia europea. The third things that we studied in order to justify the resistance of Balanophilia europea in acidified environment is the homeostasis, the homeostatic potential. To do this, we analyze the concentration of boron isotope. There are papers in geochemistry that explain you why the concentration of boron isotopes is a proxy of pH. So the higher is the boron isotope in the skeleton, the higher is the pH in which the skeleton was formed. This is the concept. So we measure the concentration of boron isotope in the skeleton of Balanophilia europea living at different pH. You see here in the x-axis, the oceanic pH. And in the y-axis, you see the boron isotope concentration in the skeleton. And you see how it is homogeneous. There is this slope that is not significant. So there is an homogeneous concentration of boron isotope in the skeleton of Balanophilia europea. This means that being boron isotope a proxy of pH, the pH of the calcified fluid where the skeleton was formed is the same. The same is the boron isotope, the same is the pH. This means that, that's the point, the last things I want to say before having some break, the meaning is this one. Even if the oceanic pH is decreasing, you see here in the x-axis, you have the oceanic pH. The pH at the calcified fluid that we extrapolated from the concentration of boron isotope, geochemistry and mictology, remain the same. Even if the oceanic pH is decreasing, the pH in the calcifying fluid remain the same. You see this slope, but is not significant. In other words, translating that result in this concept here, in this model, means that even if the oceanic pH is decreasing, so the calcifying fluid is becoming enriched of hydrogen ions, the pH in this calcifying fluid remain constant. So this means that this calcium ATPase or whatever protein is working is able to remove from the calcifying fluid the excess of protons coming from the ocean. So this coral, the Balanophilia europea, has a very strong homeostatic capacity, as we see here in this analysis. Means that it is able to maintain the calcified fluid at the optimal pH, even if the oceanic pH is decreasing. So that's what I had for the first part. We are gonna take a 15 minutes break. Now we have other two sections, so we have another break before finishing for today. So, so what we did so far, we did an introduction, the problem of ocean acidification, some insight on the biomineralization. We saw different model from the morphology to the physiology model. Some data about these experiments in Panarea, the transplant experiment that show us that the Zox coral has something that is helping in the resistance of calcification process with acidification. When I presented the experiment, transplant experiment, I tried to stress out that there was a 3-4 months experiments was repeated in different seasons, but the effect of acidification on corals was lasting 3-4 months. So I defined that experiment, that study, a short-term study, because one thing is for the organism to be under the stress for a short period, let's say even months, and another story is when the effect is lasting the life of the organism. I mean, another thing is if the organism is living all its life under stress condition. But this make a big difference. The scale, the time scale of the study. So we also approach the problem with a long-term effects, studying the corals that are naturally living, as you see here, along the pH gradient. So along that gradient, there are calcifying organisms, not only corals, that are actually live their life in the acidified condition. They burn there, they live there, and they die there. I mean, they are organisms that stay under stress for all their life. So this is a completely different approach, and the time scale is different. So the first thing that we study, for example, on these corals here, is the population density. I hope this bar is gonna leave soon. Let's put it here. Brava. Okay, so this is very basic data on the population density. Density means the number of individuals per square meter, that here is translated in percentage of cover of the bottom. So, but it's a proxy of the number of individuals living per square meter along the gradient. Here we see Balanophila europea and Vermetus triquetere, that is a mollusk, sessile gastropods. They are both calcified organisms. And in this paper, we also study the density and other characteristic of other calcified organisms. But I want to focus on Balanophila europea. So you see in the x-axis, you see the site. From site 1, the normal pH, and to site 4, the acidified, the most extreme environment. And in the y-axis, the population density, the abundance of the population that are actually living there. So you see that at site 1, they have a higher population density. So the number of individuals per square meter is higher than in site 2, 3, and then in site 4, where we have the condition of pH of 7.4 that is considered in our model an extreme environment because it's a pH that is not expected by the end of the century, is more acidic than the most pessimistic scenario. You see how the population decreases and disappeared in Site 4. Site 4, there are no corals living there, also the vermetor triqueters. So this is a study that was published 10, 16 years ago that showed this data for these two species, and there are many inside the river. So what we did? We studied the population density that show us that the pH has an effect. This is okay. This is a very basic data. When the pH is decreasing, the acidification is decreasing, the corals decrease their abundance up to 0. So for sure, they are not happy with acidification. Okay. So you see how if you change the time scale of your experiment, your study, you need to take in consideration the time scale of the study. Okay, that's the first thing. The second thing that we did is the calcification. We study the calcification along the gradient, and the calcification is the results of other two important growth parameters that are the linear extension rate and the skeletal band density. So the net calcification rate, you see here the units is the amount of calcium carbonate precipitated per year per unit area. This is the net calcification rate, means the real calcification rate, the one is actually forming the skeleton. Then we have another growth parameter that is confounding used in literature to check if the corals or other organisms are happy or not. That is the linear extension rate. The linear extension rate is important, but it's just one of the growth parameters. The linear extension rate is the rate, the speed of extension or linear extension of the object. Of the object, I mean, in this case, of the polyps, of the coral skeletons. Okay. So is the linear increase in size of the skeleton. And you see, this is the pictures of one of the animals. How fast the animals is increasing its size. And then you have the skeletal bulk density, that is a density of the material. Here you see the unit is mass per unit volume. And the bulk density is the results. The bulk density is comprising the porosity of the material. Each material has a porosity, pores. Let's say empty space. Okay, the higher is the amount of empty space inside the material that you are interested in, the higher is the porosity, the number of pores within the material, the lower is the density of the material. Okay. So the skeletal bulk density is taking consideration the pores, okay, the number of pores of the within the material. Okay, so let's look at the calcification rate at the 3 pH, because we studied the corals naturally living there. So now we are talking about corals naturally living. So corals that are in Site 1, Site 2, and Site 3. So the net calcification of the 3 sites, you see, the net calcification is 2.2, and then is decreasing 1.9, 1.8, and this decreasing is significant. And we can plot this with this model that show how the calcification rate is decreasing with acidification. So, as we understood, when the stress is not 3 months, but is for all the life of the organism, it does have an effect. So the calcification decrease, the linear extension rate is homogeneous. So it means that at the 3 sites, after 1 year, this is an important concept, after 1 year of growth, the size of the corals is the same, but the calcification rate decreases. So if the size remains the same, but the corals has less material to calcify, mean this the consequence of this is that the skeleton must be less dense. Same size with less material, the skeleton must be less dense. The density of the material must be less than in the normal condition. In fact, here you see the skeletal bulk density that is significantly decreasing with increasing acidification. But the skeletal bulk density is a function of porosity. So if the material becomes less dense, we must have an increase in the number of pores and in the size of pores. In fact, we measure this at the physics department through nuclear magnetic resonance. We were able not only to measure the porosity, the amount of pores, but also the size of the pores. And we saw that the porosity in acidified water was increasing, but also the size of the pore. Not only more pores, but each pore was bigger in size. So this is the model showing how the porosity is increasing with acidification. So when we look with the long-term effect, we found an effect of acidification on these corals that in the short-term effect, they didn't. Still, the short-term effects tell us that this coral, this specific species, has something compared to the other that make it more resistant. So that significant remains. So this is the equation that is connecting the net calcification rate with the linear extension rate and skeletal bulk density. This was for a classical paper from Loaf and Barn on poritis, Journal Experimental Marine Biology and Ecology. That's the equation that is connecting the bulk density, the linear extension rate, with the net calcification. So basically, the net calcification, you can estimate the net calcification through this equation. Net calcification is equal the linear extension rate times the skeletal bulk density. So what we found that with increasing carbon dioxide acidification, the acidification, with increasing acidification, the net calcification rate decreases. Okay. The linear extension rate remain constant. Okay. So as I said, if the size is the same, but it has less material, the density of the skeleton must decrease. In fact, the skeletal bulk density is decreasing. So the net calcification is decreasing because of decreasing of skeletal bulk density. How the skeletal bulk density is decreasing? The skeletal bulk density is decreasing because it's an effect of an increase of porosity. So in acidified water, the corals, basically, they have the same size, but they suffer of osteoporosis. They have the skeleton that are weaker because they are enriched in the number and in the size of pores. So what is the biological significance of this acclimatization processes? I try to explain you this biological significance, how I understand it, through an anecdote, to a story, okay, that I try to involve you. Let's say that we need to build a building, okay? In normal condition, we have all the cement available. The sack, you see, the sack is full. So we are going to build a normal condition. We have a normal amount of calcium carbonate. We are going to build a normal building, a normal phenotype, okay, normal building. That is this one. Is a house that has a thick wall and a quite big room inside. Okay, that's our building, that's our house. Let's do the same story in depressed condition, depressed calcification. With depressed calcification, depressed condition, the company that they need to build the house doesn't have all the full sack, but has a half sack full of cement. So in this condition, you may have two choices. You have less material. You may have two choices. The first one is building a smaller house, maintaining the thickness of the wall. You have less material. You cannot perform like in the normal condition. You have less material. So you have a trade-off, a choose. You can decide to build a smaller house, maintaining the thickness of the wall, or, like Balanophilia does, building a house of the same size, but because it has less material, the thickness of the wall will be thin. Okay? You don't have all the material, so you need to build a house, but you have half of the cement. You build a smaller house, maintaining the thickness of the wall, or you build the same size house with a thinner at the expense of the thickness of the wall. And this is the strategy that is selected for these corals. Maintaining the linear extension rate in the depressed calcification, it maintains the linear extension rate, means that after 1 year, 2 years, 3 years, the total size, the total volume of the skeleton is the same. Okay. But because there is less material, the bulk density is decreasing and the porosity is increasing. So is maintaining the size at the expense of the strength of the skeleton. So the house, okay, the choice that Balanophilia choice, the strategy that is selected for Balanophilia is this one: building the same size at the expense of the thickness of the wall, increasing the pore space, the volume, the empty space inside the coral, inside the skeleton. So what is the gain and the loss? This is a trade-off. Of course, we have a gain and a loss in this strategy. What is the gain of maintaining the size at the expense of the bulk density, at the expense of the strength of the wall, of the building blocks? Why I need to maintain the size? The reproduction is a key role, has a key role in this strategy. Balanophilia europea is sexually obligated. It's not a colony. It doesn't know how to do budding. It cannot reproduce asexually by stem cells cloning. No. Balanophilia europea only knows how to reproduce sexually, so is sexually obligated. So it must do sexual reproduction. Sexual reproduction, that's a point very important in biology in general, because we hear mistakes all the time. Sexual reproduction needs size, not age. Is the size of sexual reproduction. The age depends on the growth rate. Can be early age, old age, depend how the organism is growing, the growth rate. But what you need for sexual reproduction is the size of the body of the organism, because sexual reproduction needs space for hosting the gonads. So this Balanophila europea is an hermaphrodite, a simultaneous hermaphrodite. And you see here the morphological picture showing you the oogenesis and the spermatogenesis. So basically, you have here site 1, site 2, site 3, and then you have the developmental stage of the oocyte, early, intermediate, and late, full oocyte ready for reproduction. These specimens were collected at the same time at the 3 sites. So you can see how the early stage of oogenesis is exactly the same at the 3 sites. Then you have the intermediate stage, and you can see the nucleus moving from the inner part of the oocyte to the periphery, toward the periphery. And this is at the same time, we have the same developmental stage of oocyte at the 3 sites. And then you have the full developed oocyte, where you can see the nucleus that is moving to the periphery, very close to the cellular membrane, ready to be fertilized. And you have this exactly the same pattern at the same time at the 3 sites. And you have the same analysis for the spermatogenesis. This is again site 1, site 2, site 3. This is the early stage of spermary or tests. Here in corals, we talk about spermary. The spermary maturation stage, the early maturation stage, intermediate, and the full maturation stage of spermary here, where you can see all the dots. The little dots are the test of the head. Sorry, the head of the spermatozoa. And the lumen here, the pink area here, is where all the tails are oriented. So the spermary have this specific bouquet morphology that is exactly the same at the 3 sites. So this is a proof that the corals maintain the size. They reach the sexual reproduction at the same moment, and they are reproducing at the 3 sites. Even in the more acidified condition, they are able to reproduce. We also have a quantitative analysis of the spermatogenesis and the oogenesis. You have here the size class of the oocytes. A site 1, site 2, a site 3, the corals well collected at the same time, and you see how the size frequency distribution of the oocyte is actually overlapping. Means that the population of oocyte is actually at the same stages, and the same is for the spermary development. You have here the maturation stage from 1 to 5, and you see how the distribution of maturation stage is actually the same at the 3 sites. So the corals maintaining the extension rate, the gain is the maintenance of the sexual reproduction activity. If they would choose the other strategy, The other strategy would imply a decreasing size and a not possibility for sexual reproduction. So the other choice is not evolutionary useful because they will not reach the size of sexual reproduction. In this way, they reproduce the size of sexual reproduction. What is the loss of this strategy is the loss of mechanical strength. The skeletons are porous, much more porous than in normal condition, means that they are weaker, they can be more easily fragmented, they can be more easily attacked by parasites from endolytic excavator, endolytic organism. So this loss of mechanical strength is probably one of the cause of the decreasing in population density that we have along the pH gradient. But we also have good news here. After many bad news concerning climate change, maybe here we can have also a good signal, because in this graph, this is again caldera oceanography. You see for the latitude of 38 degree north, that is the latitude where is Panarea. You see the oceanic pH before the Industrial Revolution, today, and the projection for the end of the century, different IPCC scenario. And you see that for the latitude of Panarea, in the most pessimistic scenario, which is A2, the minimum pH expected by the end of the century is around 7.7. The intermediate pH is 7.9. So look at the graph on the right. You can see how at 7.7, the most pessimistic scenario, the corals are leaving. And of course, also at the intermediate scenario, the corals are leaving. So corals in Panarea at pH 7.9 and 7.7, that are the one predicted by the end of the century by different scenarios, they are actually leaving. They have a weaker skeleton, of course, because it's more porous than the normal condition, but they reproduce. They are still able to reproduce, sexually reproduce, as I demonstrated. And what is the good signal here is what I mentioned you already at the beginning of this section. They disappeared at 7.4. They only disappear when the pH dropped down to 7.4. That is not realistic. 7.4 is a pH that is more extreme than the worst case scenario. The worst case scenario is 7.7. 7.4 is down here, is not in the prediction. So they only disappear when in this extreme condition that are not predicted by the end of the century. Instead, for the condition predicted from the different scenarios, 7.9, 7 there, they are living, they are reproducing. So they only disappear in a condition that is worse than the predicted by the worst case scenario of the IPCC. Okay, now let's have another 10 minutes break, and then we finish with the very last section. Thanks. Cosa?
C'è una...
Okay, I would like to restart with the last section. Yeah, if you close the door, thanks. Okay, now the last section is again a continuation of the acclimatization story of this Balanophilia europea, these corals, these Zox corals in along the gradient. So we arrived here, okay? So basically we found that the corals decrease the population density with acidification, and in terms of calcification is decreasing the calcification, but is maintaining the linear extension rate at the expense of the bulk density that decrease with increasing porosity. And we try to understand what is the gain and the loss. The gain is the maintaining of sexual reproduction that need space. Okay, the gonads, especially the ovary, also the test and the embryo, they need space inside the animal. So the gain of maintaining the linear extension rate is the size, is the maintaining the reproduction. The loss, this trade-off is the loss of mechanical strength. That is partially explaining the decreasing in population density. Now, this is a piece of adesso, grazie. The number of cells. So I was saying the gastroderming is becoming hypertrophic, more thick, okay, and this is due to the increase in number of ZOCs hosted by the animal cell. You see here the number of cell per unit area in site 1, site 2, site 3 how it increased from 5,000, these are the number, to 10,000. So the number is increasing of 100%. The population density of symbionts is increasing by a factor of 100%. So more algae. Then you see here, this is the concentration of chlorophyll per unit, per cell, per ZOX, per single algal cell. And you see the chlorophyll is homogeneous. And this is in agreement with the fact that photosynthetic efficiency of each cell is the same. Okay, so the results of this story is that in the more acidic side, where we have much more chlorophyll, pigments catching light, than in the normal condition. So we call this in ecology carrying capacity. The carrying capacity is the amount of individuals that can be supported by the energy of the ecosystem. And you see that with higher carbon dioxide in the environment, the number of cells that can be hosted is increasing. So basically, we have an increase in carrying capacity. More cells, more algal cells within the cytoplasm of the gastrodermis. So more photosynthesis. More cells, more pigments, more photosynthesis. Okay? So the actual amounts of carbohydrate synthesized by the photosynthesis is increasing in the more acidified sites, in the hypercapnic or higher carbon dioxide environment. So the corals have more energy. In the acidified environment. And this is another thing that is having a bit of impact in the people that are interested in these stories, because this is thanks to Todd Lajeunesse here. This is the taxonomist of the ZOX, is an authority in the taxonomy of the ZOX. And he made this phylogenetic analysis, this genetic distance among the ZOX of the three sites, and he found that. So first of all, we collected the corals not only from site 1, site 2, and site 3, but corals also from around Italy. Okay. In normal condition. So, for example, we collected the corals from Elba Island, from Croatia, from Puglia, from Pantelleria, from Calabria, in normal condition, like the condition of site 1. Okay, and then, of course, we collected the corals at site 1, site 2, and site 3. And you see here the genetic distance. What does it mean, this story? It means that the corals living in site 1 in Panarea, the ZOX living in symbiosis in site 1, the site 1 ZOX, let's say this, the site 1 ZOX are more similar to the ZOX of all the other sites around Italy where the normal conditions are, like Croatia, Elba, and the other sites, than the ZOX of site 2 and site 3. Okay, so again, another way to say the same story, the ZOX of site 1 are more similar to the ZOX of Croatia, that is thousands of kilometers away, than the ZOX living in site 2 and site 3 in the acidified environment. So ZOX living in normal condition are all similar. ZOX living in acidified condition, they are genetically different. According to Todd analysis, they all belong to the same species, that Philozoon balanophilium, that is the dinoflagellate species, but the genetic of the site 2 and site 3 Philozoon balanophilium is very different from the other one. could be a subclade, a subspecies, a sub-something. We don't know, but they are different. So not only we have more algal cells, okay, we have more algal cells of a different clade, of a different genetic group. Maybe zocs that are adapted. We can use adaptation in this case because zocs are living in different environment million of years. So probably we have more zocs of adapted genotype. And then we measure the carbon to nitrogen ratio. We need to, well, that's the result that we got. We also involved Jonathan Harris in some of the explanation of this pattern, even if he is not in the author, is the acknowledgement of this paper. Jonathan Harris, I mentioned because he's going to have lectures on Friday here. So what we did here, we did the carbon to nitrogen ratio. I know there are students working on carbon to nitrogen ratio, and you know that this carbon to nitrogen ratio is able to give up some information about the trophic strategy of an organism. Okay, about the trophic strategy of the organism. We were able to measure the carbon to nitrogen ratio in two separate compartments, the tissue compartment, that is the orange, and the algal symbiont. We separate by centrifugation the zocs, and so we got the zocs compartment from the rest of the animal tissue. Okay, that's possible. So we have two compartments, zocs compartment, no, and the animal tissue. They were together, we separated. And then we measure the carbon to nitrogen ratio in the two compartments. And let's start from the ratio in the tissue. You see the carbon to nitrogen ratio that is starting around 6. That means that in the tissue there are 6 carbon per 1 nitrogen atoms. And this is more or less a normal ratio also in other organisms, okay? But then this normal ratio is changing crazily because from 6 goes to 0. Okay, this means that the denominator of the ratio is increasing exponentially. So why the denominator, why the nitrogen, and how the nitrogen is increasing so much in the tissue of the corals? Together with the collaboration in another paper with Marco Candela group that is going to have his lecture Thursday afternoon, we found that corals living in site 2 and site 3 are hosting in the macrobiome, so in the bacteria, you know, you have a hologenomics lecture, yes. In the macrobiome, all the bacteria associated with the corals, we have bacteria that are performing nitrogen fixation. What is the nitrogen fixation? You know, you are all you have these bases. Just to remind the meaning, the nitrogen fixation is the process by which bacteria, microorganisms called diazotroph, they mineralize the atmospheric nitrogen through this equation that you see here. They take atmospheric nitrogen that is also dissolved in the water, okay, because there's equilibrium before in the atmosphere, in the water. They take the nitrogen from the environment, the atmospheric nitrogen, and they convert the atmospheric nitrogen in mineral nitrogen in ammonia that you see here through a very expensive chemical reaction, because for 1 molecules of nitrogen, of atmospheric nitrogen, they need 16 ATP, okay, for producing 1 molecules of nitrogen, of ammonia. Okay, so it's a very expensive process. When they take all this energy, probably they also competing with the corals from the respiration, from the ATP deriving from the respiration of all the sugars coming from the photosynthesis. Okay.
So those that happen in the cold?
In the corals, yes. In the microbiome of the corals, inside the 2 and 3, there is a strong nitrogen fixation activity. Okay, so I was stressing, so I cannot go into the. Thank you, Jeff, for entering in this nitrogen fixation. It costs a lot of energy. You see, 16 ATP is a lot of energy. The question is where these bacteria are taking all this energy in this system. Probably part of the sugar that derive from the photosynthesis that is so strong is also converted in respiration, so in ATP that is used by the bacteria. So the bacteria are mineralizing the nitrogen in ammonia, and ammonia, as you know, is essential for the growth of which kind of organism? Which kind of organism needs ammonia for growing? No, it's a general question on Earth. Who's using ammonia? Non l'ho sentito. Can you repeat? On Earth. Is autotroph or heterotroph organism? Who's gonna what's the in the food chain? Who's going to make the nitrogen, the mineral nitrogen, organic nitrogen? Algal bacteria, all the plants. All the plants. They need water, carbon dioxide for the photosynthesis, but they also need nitrogen for the production of their own amino acids. So plants, the mineral nitrogen, the ammonia, it's a fertilizer for plants. When the agriculture they fertilize for the production of crops, they put nitrogen. So nitrogen is an essential nutrient for plants, for autotrophic organisms. So probably this nitrogen here is also part of the carrying capacity of the algal cells. Alga, photosynthetic organisms, like all the plants, they need nitrogen. Okay, so this nitrogen is also contributing to the carrying capacity, to the increasing in the population of these cells. These cells, they cannot live. The algal cells, the plants, they cannot live without nutrients, inorganic nutrients like phosphorus, like nitrogen. So that's another point. So there is a loop. Okay, the algal cells give ATP for this reaction, but they obtain nitrogen for their own growth. So it's a ecosystem loop there in the corals. There is. Then what this diazotroph do with all this nitrogen? They also produce amino acids. They are photosynthetic organisms. They are photosynthetic, I want to say, autotrophic organisms, and they use this ammonia, okay, to produce amino acid, like the glutamine is the one amino acid. So in this site 2 and site 3, we have nitrogen fixation, means that this diazotroph are producing organic nitrogen amino acid starting from the atmospheric nitrogen. They're producing amino acids. So this increase in organic nitrogen is increasing the denominator of this ratio, leading to a carbon to nitrogen ratio that is almost zero. According to people that study this problem, the only explanation that we can have And we prove actually the explanation that we prove in this paper was the nitrogen fixation. And this paper that is cited here together with Marco Candela, we prove the presence of this strong nitrogen fixation activity in site 2 and site 3. And then we have in the algal compartment, the symbiont, the increase of carbon to nitrogen. So it means that the carbon is increasing. And why is increasing? Because as we said already before, the carbon is increasing because there is the fixation of the carbon, because there is more photosynthesis. So this increasing is because the increasing in carbon to nitrogen in the algal is because there is more photosynthesis. The decreasing in carbon to nitrogen ratio, the decreasing is because of nitrogen fixation in corals, in the corals. Now, the very last part of my story, in 10 minutes, I guess I finished. So again, the homeostatic is the last part that is also the most complicated, probably in the last 10 minutes. Complicated to make it easy. So I want to restart from here, from the homeostatic, the homeostasis. This is the pH at the calcifying fluid that is homogeneous, even if the ocean become more acidic. The ocean become more acidic, more proton enter into the calcifying fluid. Jonathan thinks that is seawater, but that entering, okay. So the ocean is more acidic, more hydrogen ions, it's probable that they will enter the calcifying fluid, but the corals are able to remove this excess of hydrogen through the work of the calcium ATPase pump or other pump, where they take all this energy from the increased photosynthesis that we just saw. So there's more energy, okay, for the system inside 2 and inside 3. This this boron isotope derived pH was through laser ablation. There are some of you that is interested in laser ablation. These are people that are, this guy is the one who did the laser ablation in his lab. Okay, so the corals are able to have an homeostatic environment to regulate the pH of the calcifying fluid. We see here. If the pH is homogeneous, in the calcifying fluid, the pH is homogeneous, we expect what? That the omega, which is the saturation state of the carbonates, in the calcifying fluid should be the same. If the pH is the same, the omega, we expect that is the same. In fact, that's the omega. The omega is homogeneous. Okay, so the ocean become more acidic. The pH at the calcifying fluid, where the calcification rate take place, is homogeneous, and the omega at the calcifying fluid is homogeneous. The expectation, okay, because the omega is homogeneous, now I try to make this question to you. If omega is homogeneous, you expect that the calcification rate, the potential calcification rate, is homogeneous or is variable? Qualcuno ha detto qualcosa? We said that before, we start with this equation. We said that in theory, from a thermodynamic point of view, is omega is high, okay, the calcification is favor. Is omega is low, the calcification is endangered. Okay, so calcification potential that we define gross calcification, the calcification potential from a pure chemical point of view is dependent on omega. So again, if the omega is constant, you expect the potential calcification is varying or is constant? It's constant. That's expectation. In fact, the potential calcification that we call gross calcification is the same. That's the gross calcification, the three sides. The gross calcification. But you know that the net calcification, the one is actually precipitating and is forming the skeleton, is decreasing. So we have a delta here. We have the potential calcification that is homogeneous. Okay, but the net calcification, which is the calcium carbonate that is actually going to form the skeleton, is decreasing. So there is a delta. So I imagine this like a snow, starting from the top, snow of calcium carbonate that it starts, but then the calcium carbonate that actually settle on the floor and forming the skeleton is less. So there is a loss of calcium carbonate. The gross calcification is the same, the net calcification is decreasing. There is a loss of calcium carbonate. Where does calcium carbonate go and why? So you know this equation, I explained you before. There is another equation, this one, that is relating the net calcification to gross calcification and dissolution. This equation that is published in this Nature paper say that the net calcification is equal the gross calcification, so the potential calcification, minus the dissolution. So if the dissolution is 0, you have the net calcification, the gross calcification that are the same. But if the dissolution has a positive value, the net calcification is less than the gross calcification. So you have a delta. So part of this delta, okay, this loss of calcium carbonate may be to dissolution. The other fact that is decreasing the net calcification, as you know already, is the increase in porosity. So now the very last slide is the model, the theoretical model that we published already a few years ago. We are updating this model when we get the new results. It's the last things I'm going to say. Basically, this model is summarizing the result that we got so far from the Panarea acclimatization study. So we are comparing the characteristic of site 1 and site 3. Site 1 is the control site. Site 3 is the more acidic site where the corals are living. We have different compartments. Okay. We have the seawater, the ocean. Then we have the calicoblastic cells as a second compartment with the calcium TPAs or whatever pump it is. Chemistry of the calcifying fluid and the phenotype, the skeletal phenotype. So let's compare site 1 and site 3. And the main question is here, how the porosity is formed. Why and how the corals make pores in their skeleton. Must be a biological control mechanism depending on something, and then I try to understand how we explain this in this theory. Okay, so let's compare the ocean. In site 1, we have a pH of 8.1, Okay, and the omega is around 4. In site 3, the most acidic site, we have a pH of 7.7, and the omega is half of the omega of the control site. So this is an environment that has a lower omega. So for sure, because that's the thermodynamic, for sure, the classification rate, the classification there is more difficult. That's a thermodynamic. It's more difficult to calcify here than the normal condition. Then we have the calicoblastic cell. The calicoblastic cells are the cells that have this calcium ATPase pump. And you see the calcium ATPase working in site 1 and in site 3. And you can see by the number of arrows that we put in the model how the calcium ATPase at site 3 must work harder than the site 1. Why? You see here the chemistry of calcified fluid. The chemistry of calcified fluid is homogeneous, is 8.3. You see the statistical the confidence here, you see that are overlapping. There's no significant difference in pH in the calcified fluid. And the same for the omega. The omega, the calcified fluid is the same. So why the calcium ATPase are working harder in site 3 compared to site 1? Because they need to remove more protons from the calcified fluid because the ocean is more acidic. We have more protons in the ocean, more protons in the calcified fluid, and the pump must work double work than a site 1. Why double? Because the omega is half of site 1. In site 1, we have omega that is 4. In site 3, we have the omega that is 2. So you need to work double in site 3 to remove the excess of hydrogen. And they succeed. They succeed because we know that the pH of the calcifying fluid is the same and the omega ramona is the same. So they're able to maintain the optimal pH, even if the environment is more acidic. So why and how the pores are formed? Our answer to this question, this our theory, we did not perform any experiment yet, is this one. In site 3, there is more energy for the corals, for the holobiont to live, to do their metabolism. Why? Because there is more photosynthesis. There's much more photosynthesis in site 3 than in site 1. So there is much higher amount of carbonic, organic carbon, sugar for respiration. So there is an higher production of ATP. But this ATP, this higher amount of ATP, is not enough to activate all the calicoblastic cells. So there will be, you need to imagine the calicoblastic cells like a carpet of cell, okay? Some cells are activated because they have enough energy coming from the respiration of the higher photosynthesis, but there will be other cells that are not activated because they have not enough energy. So we imagine a carpet of activated, non-activated, activated, non-activated, activated, non-activated, and so on. So if we go back at the very beginning, and you remind how the skeleton is formed. This is the quiff theory. Ecco qua. Looks at this last line here. Let's imagine that these two cells are activated, the first two, but the next one are not, because there's not enough energy. And the next one, two are activated. So they need to do a new layer. The first two and the last two that are activated, they're going to form the skeleton. But the one in the middle that are not activated, they are not going to form nothing. So they leave a pores. So our theory is an energy-based theory for which in more acidified environment, even if the carrying capacity of the algal compartment is much higher, there is much higher carbon fixation, much higher amount of substrate for respiration and production of ATP, it is not enough for all the calicoblasts to be activated. So you have a biological control through the amount of photosynthesis of the pores formation. When the cell is activated, there is the layer. Is not activated, there is an empty space that is pore. Activated, non-activated. And if you think this for many layers, you can imagine like two columns of calcium and a big hole in the middle. In the other story, that is our big pores. I don't know if we are gonna able to ever demonstrate this, but that's what we think on the formation of pores in the acidified environment. So in conclusion, during the last 17 years, we worked on long-term study on temperature effect that I did not talk at all today. We did this long-term study along the latitudinal gradient from Genova to Pantelleria. We studied the different population along this temperature gradient of about 2 Celsius. So are naturally corals living along the Italian coast, and we found that the ZOX corals are more sensitive to temperature than the non-ZOX corals. This is probably due to a deficit in the photosynthetic process with increasing temperature. Then today I explain you the result of the short-term experiment of acidification on corals, and we found in that case that the ZOX corals are more resistant than the no-ZOX. They for sure, Bologna Europea at least has for sure something that when you stress it for a short term, make the coral more resistant. I don't know what is it, but there's something there. And then there is the long-term experiment, the long-term study that I show you this afternoon, where, of course, when the corals are under stress for all their life to the acidified condition, they have some negative effect on calcification. But there is also a positive message there, because if we look at the performance of the corals, even in the most extreme condition predicted by the IPCC, they are living there, they are reproducing, they have a weaker skeleton, but they are able to live in that condition, which is a positive message. There is a question mark because I still did not finish the work on non-ZOX corals comparable to Balanophila europea. We are now close to publishing something, so maybe in the next years I may have some information on this question mark, but for now that's what I have. So thanks for your attention. I'm done.
Thank you.
Welcome. If there are any questions, I'm happy to try to answer. No. No questions, so
You think calcification is energy limit? It's because my impression is that we produce sugar in enormous amounts, but
are becoming much more autotrophic because they increase the amount of carbon fixation. They also increase the amount of amino acid, at least in the microbiome compartments. So the holobionts become more autotrophic in the acidified environment. He has more energy, more substrate for respiration, more ATP, but yes, our interpretation of the porosity is that it's because the calcification is energy limited. Yes. Thanks.
So where food availability acidic sites?
No.
The same as that.
Thanks for the question. Temperature, nutrients, so inorganic nutrients, which is sustaining the photosynthesis, the phytoplankton and zooplankton, are the same along the gradient. For example, the average temperature, the annual average temperature is the same because they are the same depth and the distance is only 35 meters. So the only things that is changing from among the side is the pH. Nutrients are the same. Pressure, salinity, and no, temperature I already said. Yes, temperature is the same. Pressure and salinity is the same.
So
Pressure and salinity because is affecting omega. Okay, but are the same. It's the same temperature, it's the same pressure, it is the same salinity. So the only things that is really changing in this system is the pH. So there's no more predation in this. It's just that was your question.
yeah. Be disturbed by the acidic and
By the emission,
by the CO2 emission.
we don't have this evidence, but it's a good point because no, we measure the nutrients, all the things that, but the zooplankton community, the abundance in the water column was not measured. That's a good suggestion, please.
going back to the fish model to explain the porosities. Could it be demonstrated by doing some histological section on the chemical-blast area to see whether there are some cells with team and some other not? You know, if you have been watching cells, some of them actively gets fine, some of them not gets fine. It should be possible to see the
Really?
right?
You
know, if you make cross sections and maybe do some, I always or some
histology, maybe also other kind of microscopic morphological approach. Yes, that's a good suggestion. We didn't think about that so far. So you think that we may be able to see like the ectoderm with like a skeleton, no skeleton, skeleton, no skeleton.
Marker for one of these proteins. Maybe a marker will be very well expressed in some cells, in some chemical
Oh, wow.
and not at all in some others. And maybe,
That's a good.
so then this we go then the same direction and say what you.
Yes, nice. Thanks. Okay. So now it's please. No, I don't want to force you here. So if you, it's a
Well, during by the mollusk model, you know, when mollusks work in anaerobiosis, they tend to resolve slightly the inner surface of the shell. So that because they produce a lot of acids, and they immobilize some calcium. Could it be the same scenario with your balanifier coral that actually at the interface with the chemical vesting epithelium of the strong skeleton, that for some reason at low pH we may have somehow some anaerobiosis and some research, meaning that the coral tend to make its function or the positive calcium carbonate, but somehow there are some secondary mechanisms which
secondary mechanism.
resolve the calcium, the uniform calcium. It might be possible, or this just came in my head that because of this.
If you dissolve in anaerobiosis, with in an environment with low oxygen, you mean?
Yeah.
The mollusks, they dissolve.
They can resolve slightly, but it's something that happens for some, especially in mussels, for example.
What is the explanation of these things in mollusks? Why they do this? Why it's
some acids. Well, I don't know the details of that.
Right.
There is a acid production at the interface between the mantle of the shell, and then you have the slight dissolution. And maybe the animals could take the profit from that to reabsorb some calcium for some use. So I was wondering, maybe some corals somehow.
Anaerobiosis, no respiration.
It's just
Yes, I need to think about that.
just
Because no oxygen, there's no respiration. No respiration means that the pH should increase, because the pH decreases with respiration. So I don't really catch the link between anaerobiosis and dissolution. I'm missing something, but maybe later when we are drinking, I'm sure we will find a solution for that.
So the porosity, so are the pores, so are these spaces distributed? So because if the coral is using such kind of mechanism, say calcium, it must be evenly spaced. If it's not the case, then there's no those growth process. You understand my question? So you would expect that make pores?
Pores, yes.
That they need to have a kind of almost equal distance from each other. To have still more or less stable skeleton. if you made very large pores and a very large distance in all pore, it doesn't have much sense. So you would expect an even distribution.
Increase in the size of the pore, you ask,
The space,
the spacing.
that's what we would expect, but the pore is all.
Ah, so I don't know. I mean, we about the porosity, we know the amount of pores, and through the magnetic resonance, we know also the size distribution of the pores. And what we found, I just mentioned porosity, but what we found is that it's increasing the amount of macropores. We divide the distribution of pores in micro nanoporosity and macroporosity, because the NMR give you a distribution, and you can from the distribution you can divide the size class for. So you have at least two macro size distribution. The micro pores, the micro and macro, micro porosity and macro porosity. Now I don't remember the size of the pore, the cutting point, but what we found is that it's increasing the amount of large pores. So the amount of microporosity is the same. If you look at the amount of macropores, those are the one are increasing in their abundance. So the corals, because that's the reason why I increased the size of the pore there. It's not just an increasing in number, but also in size.
Maybe the mention was microchip distance in the market.
That's another PhD project. Yes. You know, you now I give you a question. The exchange with the environment, that is an acidic environment, so it's corrosive. Okay, is larger with the micro porosity or with macro porosity? Who gives the higher surface of exchange with the environment? Because these are pores that are connected to the environment. It's not internal porosity, but is connected to the environment. Okay, we are not able to measure the pores within the calcium carbonate crystal or between the crystal. Are empty space connected to the environment. So you have a higher exchange with the microporosity or with the macroporosity. The micro is order of magnitude higher, the surface. So they increase the porosity, but are limiting the exchange with the environment, with the corrosive environment, making large pore. So this is it's if you have a corrosive environment and you need the pores because the calcification is not sustained, it's better if you do large pore than the little pores. Because with large pores, you protect more the internal environment from the corrosive environment.
Chemical stability. It could have consequences for the biomechanical stability of this certain point of collapse.
In fact, they disappeared at some point. So it's a strategy that has its limits. The limits are 7.3, the 7.7. Below So that's a story. Okay, so thanks for your attention. See you tomorrow. Ciao.